to our event with Professor Tayfun Özçelik. We are very happy to see you all here. Let me briefly explain how today's event will proceed. After my short introduction of Professor, we will give the stage to him and listen to his interesting stories and advices to us as young scientists. Later, then we start with question and answer session, Q&A session, where you will be able to ask your questions to Professor. And also after the Q&A session, we will make another Q&A session, but in a blitz format, where we will ask short questions, and within a short time, Professor will give us only one single answer, yes or no, this or that. This is our general plan. Well, we planned for 1 hour, but discussing some topics with Hocam will take, I guess, more than an hour. But we will try to make it as compact and as dense as possible. Okay, then let me introduce Dr. Özçelik, Tayfun Hocam. He is the Dean of our department, our faculty, and chair of the department. He graduated from Istanbul Medical School in 1986. After that, he did his several postdoctoral research at LMU at München, then at Yale University, Stanford University, and Howard Hughes Medical Institute. Yes, Institute. After returning back to Turkey in 1992, he joined the department by first becoming an assistant professor, then associate professor, and from 2012 he is dean of our faculty. So about his research, I guess it would be better if he talks about it more and in a more correct way. So with all due respect to Hocam, we will let him to the stage. Invite.
Thank you. So just in time, coming please. It is wonderful to see your faces, and I hope it's going to be a interesting session. And while I'm talking also, if you have any questions, please don't interrupt. Seeing your bright faces, bright eyes is always the biggest happiness for me. And seeing you also dress up, thank you. And actually, it reminded me a story. I wasn't prepared, but let me start it like that. You know the Chinese culture? Yes. You know the spit cups in Chinese culture? Someone asked, huh?
I know of it, but I don't remember the story. My dad told me when I was very young.
Yeah, tükürük kopyası.
And
In Turkish, the spit cups. Now, actually, in the Chinese culture, it is, respect for the individual who is in front of you to first clean your throat, but, dramatically. That's what I was told. I read an article in Time, many years ago. Gorbachev. Who is Gorbachev?
The last leader of the Soviets?
Soviets, yes. Is visiting China. And his plane comes. And he descends down. And the Chinese are horrified. They are, really, çok yaptı, üzülüyorlar yani. They are insulted. Why? Because Gorbachev is wearing a gray suit with brown shoes. You know, this morning it was dark, and I thought I would pull out the gray pants, but it's a brown pants with a navy jacket. So, forgive me. And what happened is, you know, in such high level talks, actually, the split cups are not part of the procedure. But the Chinese got their revenge. So, during the official talks, if I'm not mistaken, Deng Xiaoping, the Chinese leader, used it. It was a sign of respect from his cultural point of view. But from a general point of view, of course, such an unusual act. So, I received some questions. Kerem sent me yesterday, and I went over them. And I think I can talk about these, all of them. I'll try to be, and you together with Islam John.
Yes, Samet was going to ask you the questions one by one. Samet?
Yeah, I can do that actually. The plan was I was going to introduce you to the questions and then maybe you can answer them. But if you want, you can just read them
Since
as well.
Hocam is prepared to the questions, you can, and you have list of them, you can continue.
I planned to organize this event based on your questions here.
Okay, thank you.
So, first of all, what inspired you to pursue a career in genetics, and how did your academic journey shape your current research interests? I mean, I think I have a very classic answer to this question. My high school teacher. Actually, many years later, I'm a graduate of Kadıköy Marif Koleji at that time, but now it is called Kadıköy Anadolu Lisesi. And there I had a brilliant teacher, biology teacher. And she introduced me to the subject of DNA. I remember vividly, as a high school student, mesmerized about DNA. At the same time, my family's friend, Altan Günalp, I don't think any one of you know him, but let's remember him also. He passed away, unfortunately. Altan Günalp was a student of Jim Watson. And he did his postdoc in his lab and came back to Ankara to establish the first department of genetics in Turkey. At that time it was called medical biology. And he was a very good friend of my parents. And he gave me the book written by Watson, and I read it. You know, this is the very famous book that he wrote, and that's how I was hooked up. I mean, I was infected with this curiosity of genetics at that time. And let me tell you one thing. Maybe many people don't know about this. Ankara has a special place in Jim Watson's life. What happened is, you know, he has many books on science and also science policy and also how to be a successful scientist and so on. And I met with him in 2010. think, 8. This was a meeting in Brno. Brno is a city in Czech Republic. This is where actually Mendel did his classical experiments. It was a meeting of the European Society of Human Genetics. And this was an ancillary meeting, I mean a side meeting, about history of genetics. And I had a presentation there. My wife, Barış knows, is interested in genome art. And I was giving a talk about science and art, the interaction between science and art in the Abbey, Mendel's Abbey. And at the end of the talk, a lady came and said, "Oh, I am Dr. Watson's associate aide, and I wish he listened to your lecture." And my wife said, "Oh, then one of those science art pieces that she produced, it was actually a scarf, gave it to her to present it to Dr. Watson." Good. Then, a few weeks later, I received a letter. Dr. Watson is inviting my wife and myself to Cold Spring Harbor. Bakın unutmayın, kalpten kalbe yol vardır. Okay? Even if I write the most interesting paper in Nature, I would get his attention. But this apparently affected him, and he invited me and my wife, and we are staying, overnight with him. Pijamalıyız. And during this trip, I learned from him that the story of his marriage, Elizabeth Watson, also there, it was actually a difficult year. Watson married quite late, not a young boy, he got the Nobel Prize. And after actually, Watson's father was also living with this family, with the young couple. And they had no bedroom, because the bedroom was father, and their house is so small. He's a Nobel laureate, but he has such a small house. So he and his wife had to sleep at the veranda. And at the end of the summer, he passed away, the father. And this was the time for their honeymoon. And they came to Ankara for the honeymoon. So, very personal story. And therefore he likes Ankara, Turkey actually. He remembers the 3 names. He's a very opinionated person. I'm sure you all know how he puts his self into trouble, all the comments that he makes. So he's not a person who is shy of expressing his opinions. If they are outrageous, he doesn't care. He told me there are 3 Turks that he admires. Number 1, you can all guess, I think, anyone who knows history, Atatürk. And he knows it very well. All the intricacies. how army, power, a monarchy, the relationship from an international point of view, on the Ottoman Empire, how it is, all these details, he's aware. Number two, our founder, İhsan Doğramacı, as a remarkable individual. And number three, Erdal İnönü, who passed away also. So, to answer your first question, which took me 17 minutes, this is my answer. What were the biggest challenges you faced throughout your career and how did you overcome them? Okay, let me try to be shorter here. Number one, after I graduated from the university and went to Yale, I had no understanding. I didn't speak the language of genomics. I didn't speak the language of genetics. I mean, in a university, especially my background is medical school, the thinking, logic is different. It is phenomenology, the effect of X on Y. If you treat the patient with this drug, this is the outcome. The heart rate is crooked, inotropy, chronotropy, and the sympathetic system does that, parasympathetic system does that. There is a phenomenological thinking. Genetics is not like that. And in order to understand that, you need to first speak the language, then, I mean, without a vocabulary, you don't speak the language. So, I knew that I know nothing, because I worked very hard, night and day, reading books. Then, at night, I would read the book, and I understand it, I understand it. No, I don't understand. And I would say, if I hit my head to the wall, things find their place, and then I understand. So, and there is always a resistance period. Then time comes, and then you start speaking. So, this was, I think, when I look back, one of the biggest challenges. Number two is a little bit darker, academic jealousy. I have to be honest with you guys. Jealousy is a very powerful feeling, and it exists in the form of academic jealousy as well. So, be prepared and don't take it lightly, but at the same time, patience is very important. Okay? Sabreden derviş, muradına ermiş. Looking back at your career, is there anything you would have done differently or any risk you wish you had taken? My short answer is no. I mean, honestly, no. I would have not done anything differently. I think this is a big statement, I understand that. But honestly, I think when I look at the turning points here, first of all, going abroad, leaving my country and going abroad to Germany, 11. I am glad I made that decision. Then I changed my field from coronary physiology and to become a cardiologist. I became a human geneticist. And this was also I think in my mind, a calculated decision. It was not a, random decision. I went to Yale for my medical science training program. Was the one that I enrolled to. It was 75% lab work, 25% patient responsibility. It was such a program. Then it continued at Stanford University, at the Howard Hughes Medical Institute. We were the first board eligible. There are specialties, subspecialties, and there are board certifications for in the medical field. My field is background is that way. So they were the first group that was eligible for board certification in clinical molecular genetics. And sure enough, there was big need. So even without applying for a job, universities were headhunting us to recruit as assistant professors. And it was a time for me, again, a bifurcation time, to come back home or not to come back home. And I chose to come back home. And I'm glad I did it. Every day, when I think of it, I'm so happy. Also, coming back to home, I was in Istanbul first, Istanbul Medical School, in the genetics department. Then and it is very unusual for someone, and my ancestors are from Istanbul as far as we can go back. My father's side is from Dardanelles, Çanakkale. My mother's side is from Beşiktaş, Ihlamur. And we are from everyone. It was I think, difficult or not an easy decision to come to Ankara. But I came, I looked, I saw, I contemplated. Then my wife came. I mean, her parents think that crossing the Bosphorus to the Asian side is going to another country. I mean, they are so attached to their neighborhood of the European side of Istanbul. But she's grown up in Istanbul at Robert College. She lived at Bebek, one of the nicest parts of Istanbul. But she said, Tayfun, this is correct decision for me. So, ever since, now it's coming close to 30 years that we live in Ankara, and we enjoy it. We like Istanbul still, it's our hometown. I always feel like Kartal, Pendik, Bilkent. This is how I feel, really. But I'm so glad to be in an environment where students like you, this is really what makes the difference. Bilkent is different. And your presence is enlightening our lives. Truly. I'm not complimenting. This is the reality. So, that's it. What do you think are the most exciting developments in genetics that we can expect in the next decade? This is a difficult question. I mean, let me first say that you can't ask Columbus to discover America. I mean, the research itself is unpredictable. What's going to lead to? I mean, it's a revelation that you make. You understand. Groundbreaking discoveries are things that you are not aware of. Otherwise, it would not be a discovery. And the future is shaped by such discoveries. So, my answer would be, my conscious answer is, of course, I don't know. In 2008, when the human genome was sequenced in an individual, 2006 actually, and you know the story. The Human Genome Project was conducted between 1993 and 2000, up to 3, let's say. 1989, I'm sorry. And the initial human genome is a compilation of individuals. The first human genome, the chromosomes were divided between research groups, some in, many in US, some in England, and so on. And at the time, actually, it was also a discussion point that I maybe told you, some of my students. Whose genome are we going to sequence? And some said, we're going to sequence the person who donates the highest amount of funds for us. Some said, important person. They even said, should we sequence Abraham Lincoln's, for example, because he was assassinated, as you know, in a theater, and his shirt with the blood is there, so it is possible to clone his genome in a environment, in a bacterial environment, to amplify it and distribute it to everyone in the genetics community. And that's some said national pride. No, my leader is more important. Yours is important. Mine is important. At the end, what happened is a group of people. And this group they randomly chose a town in US. And they randomly chose, after of course convincing the people, informed consent, they sampled a group of male and a group of female individuals. And the project was done on this small cohort. But in 2006, when a single individual can be sequenced in one reaction, this became technically feasible. Of course, the community started discussing who to sequence. And the ethicists said, be careful. You know what you are doing? Because when you sequence a single individual, you will have information on all of his genetic constitution. What if you find a predisposition to cancer? Will you talk about it? What if you find a predisposition to a late onset disease, like Alzheimer's? Will you tell us? Which parts of the information are you going to hide, filter out? Which are, and they found the solution. Watson said, I am over 80 years old now. I have a Nobel Prize. Everyone knows that I speak my mind. I didn't use other words. I'm crazy. Or not included or anything. If you find a gene for mental retardation, if you find a gene for this and that, and I'm over 80, cancer gene, whatever, it doesn't matter. So, and everybody said, yeah, I think we should do that. So this is how the first genome was sequenced. When this genome was sequenced, then the discussion in the scientific society started. Your question. Now that we are capable of sequencing a single human genome, and remember, the Human Genome Project was 3 billion. Watson genome was like 6-7 million, from billion to million, and in the same time period, at that time, it became clear that a single genome can be sequenced for $1,000. So, sequencing is no longer a research topic. It is democratized. Remember, I mean, if you live in the 17th century, you can only hear at a concert hall what Mozart plays. You don't have the luxury of listening in your home, Mozart. But recordings, democratized. Everyone is able to listen any beautiful song, any beautiful aria, when they wish to listen. Just like that, human genome sequencing is now not a utopic subject, but a reality. Nature, at that time, decided to ask 50 geneticists around the world to look into the future. And among those geneticists, they also called me and invited to write a paper. You can see this. I shared it with my students, actually. Question of the year. What would you do now that you have the technical capability of screening an entire genome? And my answer was, I think you should read it, but I was reading, I was listening, so when I received this invitation, actually, I couldn't believe that, you know, in those 50 people, my name is there. How come? I mean, there are all these big guys, and there I am also invited. And then my wife said, no, you're not reading it incorrectly, and this looks like a legitimate letter to you. It doesn't look like someone is joking on you. So this is reality. And at that time, when I read this invitation, we were at Monterey in California. Actually, I was visiting the Oceanographic Institute there. And on the way back, so we visited, I opened the clouds, and back into the car, we're driving back to San Francisco. Elton John was playing on the radio. Goodbye to Norma Jean. You know the song? You should. Listen. And I said to I found what I'm going to write about. Because when you write, when you submit it, if they don't like it, they may refuse. It has to be a meaningful also. Goodbye to recessive genes. My extrapolation at that time was that with the availability of the human genome sequencing as a tool in our hands, for the future, we will be able to uncover the genetic basis of recessive disorders. I'm not talking about dominant disorders because a large number of dominant disorders are due to de novo mutations. But recessive diseases, mutations are old, usually. So, from this point of view, at that time, of course, the genetic basis of recessive diseases was not known very well. But sure enough, one can extrapolate that in the coming years, these genes will be defined one by one. So then it is not also eugenics. I mean, you can then inform people what they are carrying as a recessive phenotype. You can also genotype the spouse. And if they happen to be carriers for a specific disorder, both of them, then they have reproductive choices. So I think this is at the heart of molecular and genetic medicine, which holds true today. And you know, why is genetics so important compared to other disciplines? Biochemistry, pathology, physiology, microbiology, and here is the genetics. What makes genetics different from other disciplines is it gives us, keyword, causality. Causality. It is the most difficult. Because, again, let me say something in Turkish. Deveye ne demişler? Boynunu eğ. Deve ne demiş? Nerem doğru. In a pathological state, in a cell, in an organism, molecules are all over the place. But what went wrong in the first place that led to an abnormality of that pathway, this pathway, and adjoining molecules, whatever, and everyone is now in turmoil. Titin is there, mitin is there, pitin is there. You see the molecules? We need to understand what went wrong in the first place. This gives us also the point where we can start developing remedies, meaning treatments. We can now target them, not symptomatic, but really addressing the situation at the causality level. Hence, in the future, we will see more and more targeted treatment options, meaning small molecules, other antibodies, but whatever, for the not symptomatic, but true treatment of diseases. So, from my point of view, I think in very general, I can say diagnostics and therapeutics are the two areas that molecular and genetics medicine is going to transform. It has already transformed and will continue. And don't forget, this change is not like a tsunami. where it comes, you know, in a short period of time and messes up everything. No. It is more like a şey neydi? Gel git in English. Medcezir.
Riptide.
Huh?
Riptide. Riptide hocam.
Tide. A little bit going, coming back, going, coming, but always progressing. Always progressing. Slow. If there were no limits of funding or technology, what impossible genetic questions would you want to answer? I think there are many different impossible questions that one can ask, but from my point of view, you know, at my age and also so on, you should ask questions that you can answer. I mean, at least have a chance of answering it. So you should choose carefully, wisely. Sure enough, when you look, I mean, our business is genotype-phenotype correlation, meaning understanding the genetic determinants of health problems in humans. You can do this in biochemical means. You can do, you can introduce mutations into a model organism in mouse, in Drosophila, in zebrafish, whatever, in cell lines, engineer things, and see changes. Well, we have hundreds of thousands of years of evolution behind us. And another experiment is going on. And the mutations are introduced into the genome of living organisms, and humans are not an exception. So these naturally occurring mutations have consequences, positive, negative. I mean, the positive is evolution, negative is diseases. So, if you characterize these events in the genome, then you can get really meaningful, significant answers to scientific issues. So, in this background, usually we have family studies and consanguinity as a very strong tool in our hands. Small families and consanguineous families really help us follow the segregation of chromosomes and markers so that we can pinpoint where the faulty gene is. Then you start digging, and you find out a gene. And then you start characterizing the mutations. In 21st century, we are now in 21st century, this has changed. Still, families are very well. But because of the availability of DNA sequencing, now as an industry, be careful, it was a home cooking. Now it's an industry. You get services, you know, scale economies. When it is produced a lot, then the prices go down. So, I mean, if there is a company that is providing the services for you, you don't need to, try it in your own lab. Consequently, already countries changed their policies. You know, 2012, London Olympics. Did any one of you watch anything or remember anything about, or were you too young at the time? Okay. Actually, in 2012, while all this popala and mambo jumbo about the Olympics was, I mean, I don't criticize Olympics by any means, but at the same time, Cameroon government in UK did a very smart thing. They changed their health policy and, as a country, decided to invest in genomics at the population level. Public health genomics and the UK Genome Project, UK Biobank. Others followed. Finland, Estonia, of course, United States, Biobank, Regeneron Biobank. These are big biobanks. At the onset, we also tried to do this in our country. I worked for about 2 years directly with the president's office. I prepared an 800-page long document. And since also I have responsibilities at the European institutions like the ERC or European Council, I had access to all the policy documents of different countries. Although this was enthusiastically supported in our country also, it never realized. I mean, one of our major funding agencies in Turkey adapted it as their institutional goal to start the Turkish Genome Project. But I don't want to be more detailed here, but the bottom line is, it was not realized. Of course, in the meantime, we were also doing our studies, and Koç University was also doing their studies. And we established actually the Turkish Variome, not at a national level, but as a university research endeavor level. And when we came together, these two institutions, and when we brought also Turkish DNA samples in credible universities outside of Turkey, and they were willing to cooperate with us, work with us, namely Rockefeller and Yale universities. We suddenly had a 5,000, near 5,000 individuals representing our country, which was actually a good enough sample to understand the genomic diversity of our country. But of course, biobanks are changing the business from top to bottom. I mean, now you have 500,000 people with complete medical records coupled with their genome. And very wisely, listen to this very well also. The British government said, we're not going to keep this data to ourselves. And they made the decision, we are opening it to the qualified people. Why? Because they said, this is such a treasure trove that we don't know how to exploit it either. I mean, there's so much in it. 500,000 people with all medical information, demographic information. Barış knows it much better than me. And here is the important point, the wisdom from a politician. Not a politician, but from a political standpoint. They also said, we know intelligence is not something that is only our. There are very smart people in Italy. There are very smart people in Turkey. There are very smart people in India. There are very smart people in Iran. There are all over. So we rely on the creative power of the scientific community to use this raw data for the benefit of humankind. And we applied, and we now have access to the UK biobank. I think the only group, surely in our country, but also very few people have access all over the world. So my questions for the future are actually geared towards answering or investigating, I should say, things that I cannot tackle on a family-based format. So this of course means complex phenotypes and other attributes of humans. We don't only have diseases, we can imagine other things. So this is all that I can say, and what advice would you give to students who want to pursue careers in genetic research? You must love it. In Turkish we say, her iş bir aşkla yapılırmış. Öyle, I'm gonna do, not do, no. Buraya kadar değil, buraya kadar gömüleceksiniz. Okay, you have to be in it with your whole body, whole soul. And one more thing, don't give up easily. I mean, there will be hundreds and hundreds of excuses, obstacles in front of you. Okay? And yine argo konuşayım. İçinizden söyleyin. Get lan. Tamam mı? Okay? I mean, sometimes you cannot convince. Derdini anlatamıyorsun. Deveyi, hendeyi atlamıyor. O kadar o zaman engelden diyorsun. Hadi. Okay? But also, very importantly, always, always, always think first before you do anything. Think first. Consult. İstişare edin. Think it over. Look at it from this direction, that direction. It takes 9 splits this way, another 9 that way. Bu yana doğru 9 takla atsın, bu yana doğru atsın o fikir. And then reach to your own conclusion. Don't be afraid to ask opinions from people, but put it in your pocket Look at it. You don't have to follow it. Okay, in Turkish, let me sum it up. Doğru bildiğin yolda dosdoğru gideceksin. It may have also consequences, but follow the path that you think is the correct path. Don't forget, even the smartest people and with their utmost good intentions may give you not so good advice. It may be possible that their imagination is not at the level that you are. So that's why believe in yourself first. Of course, always doğru bildiğin yolda dosdoğru git, burnunun dikine gitme. How can I translate? Follow the right way you perceive as the correct path, but don't be stubborn or with a vengeance. Don't do it with a vengeance. Okay? And also, one more thing. When insan lursa, bunun da şarkısı var. And really, God has funny ways. Sometimes, some things that you perceive as bad happen to be good for you, and some things that you perceive as good happen to be bad for you. What are your thoughts on the future of gene editing technologies like CRISPR? Where should society draw the line between therapy and enhancement? I think this is a short answer, very clear answer. Of course, CRISPR is a very promising, and already clinical trials are underway. So it is revolutionary. It's not only human health, you know. I mean, YouTube is full of videos, inspirational videos, how it can help us with, you know, animal husbandry, in botanics, and so on. The important thing here is touching the germ line is the red line. You don't touch the germ line because you take control of the evolutionary process of the species. From my sentence is not finished. Some of you are science and ethics students. From our contemporary, I use the word contemporary, ethics, moral, and practical point of view. Here, the key word is practical. Let me explain what I mean. You know, in 2018, Nana and Lulu were born in China. A Chinese scientist tapped with the germline and changed, I think, CCR5 gene, if I'm not mistaken. With good intentions, he says, to prevent AIDS infection in these children. And the entire scientific community, unequivocally, I mean consensus, he was condemned to do this. On ethical and moral grounds. Simply because by touching our germline, we are introducing a genetic variation that will be transmitted from one generation to the next generation, which is playing down. From practical, I said, because, again, Turkish, kaş yaparken göz çıkıyor. Our understanding of biology is not at a point where we can temper those genes. Practical, what happens? When you have a mutation in CCR5, then your lifespan is short. Nobody knew about this. Now everybody thought that it's a receptor that is critical on the cell surface for the attachment of the virus, HIV, human immunodeficiency virus. Yes, it is indeed that. But in biology, we have other functions for a given gene, for a given protein. And it looks like CCR5 knockout is with a shorter lifespan. So, when we are trying to do something, we shouldn't cause another thing bad. How did you introduce DNA-based identification to the Turkish judiciary system, and what impact did it have on the legal and scientific community? Okay, you know, actually, when I came back home in 1992, end of 1993, I was already aware that back home, I mean in Istanbul, I'll have to do some sort of forensic work. Because this was the time when forensics was introduced to the judiciary system for the first time in America, in European countries, and sure enough, it's not, you know, it's not a secret that there is going to be a DNA lab in Istanbul. And sure enough, someone will come and ask the services. Actually, my mentor told me this when I was coming back home. And she made the analogy, I think I told this in the human genetics class. She said, the most frequent DNA tests are done for individual identification. I mean, we are all doing disease genetics, but individual identification is very important. So, in a way, I was prepared. And sure enough, about 1 or 2 months after my lab was established, some people came, like the security people. And then another person came in, looking like a very important person. Apparently he was the Minister of Justice, Adalet Bakanı gelmiş. And together with him was the rector of the university, and this young guy who came from America, and he said, hello, my son. And yes, and then they called me to the Forensic Medicine Institute at Bilkent in Istanbul, and they said, now your duty is to introduce DNA-based individual identification in our country. So my reaction can be, you know, no, I'm a scientist. And so on. Probably they would find I'm refusing the government. Anyhow, since my mentor prepared this, I said, okay, her should be highly and we started this. So actually, this is also the reason why I came to Bilkent. Because suddenly, I became a policeman. Meaning, all the are coming to me. I mean, my lab was giving 3,000 DNA reports per year. 3,000 means 9,000 people, because in any paternity case you have three: father, the alleged father, mother, and the child. So the university was very happy because, you know, there is all these tests are, there is no return, nothing. Biz Boğaz Okulu'nda çalışıyoruz. And of course we have our, that's not the point anyway. But in the meantime, of course, I had extremely interesting cases also for our country. For example, we did the first forensic analysis from post-mortem material. I mean, exhuming a body from the grave and how to do, you know, and this which actually gave me a lot of insight into anti-DNA research technical. I had an understanding of that, through that. Some very interesting stories that I shared with my human genetics class that I don't want to go more into detail right now. And at the same time, also I trained the police lab, gendarmerie lab, and the Forensic Medical Institute staff in Turkey, all of them, for 1 year, so that it became a service for the whole country when I left. Can you share the story behind one of your most significant discoveries, such as the mapping of SNRPN gene in Prader-Willi syndrome? Okay. Now, first of all, let me reiterate, you cannot ask Columbus to discover America. So, in the very first place, when I started my research, I was not trying to discover the gene for Prader-Willi syndrome. Prader-Willi syndrome was a very, interesting disease for all the geneticists at the time, because genomic imprinting, the concept of genomic imprinting was developing as a new field, and Prader-Willi syndrome is the prime example of it. So if you find the gene for Prader-Willi syndrome, then you have a foothold in the human genome to analyze the molecular basis of imprinting. My own purpose is, together with my mentor, we have a hypothesis. This is juxtaposition of maps. This means we have a physical map of the human genome, meaning chromosome 1, 2, 3, 4, 5, 6, 7, all the chromosomes, and whatever gene has been cloned at the time. And in my time, it was like 1,500 genes were cloned at that time. You put a chromosomal locus. I mean, insulin gene is on chromosome 11, for example. Like all of these genes, DMD, Duchenne muscular dystrophy gene, is on chromosome XP21, and so on. Then, at the same time, Helen Donis Kellner started generating the first, do it fast. Do it fast. They started generating the linkage map. It's a map of recombination fractions. So it is not a physical map, but it is a mathematical map. So without knowing the gene, you can deduce the location of a disease gene. So our our central hypothesis is to juxtapose these two lengths. Meaning, chromosome 7 and gene X is on chromosome 7. And you look at the linkage map, cystic fibrosis is on the chromosome 7 and in the vicinity of the critical region. Then your gene becomes a candidate for that disorder. And our strategy was to map all the genes that people are cloning at the time, collaborate with them, and get the cDNA probes for these genes and determine the chromosomal locations. Then we can look at the linkage map and see what's going on. In this exercise, here is the important part. I mapped a gene that came to me from a collaborator, and it went to a particular human chromosome. And we do the mapping also in mouse. Now, ne oldu?
This for the GA points we have to.
We have to distribute the paper.
Okay, distribute it, don't worry. The important point is groups of synteny. Meaning, something that's on the mouse genome has a position in the human genome. Only X chromosome is in both species X is X. But something on human chromosome 17 is always human chromosome, in mouse chromosome 11, for example. And these chromosomal localizations didn't fall into this expected synteny group. There are two possibilities. We are defining a new synteny group that is not known in the human genome. Second possibility is we are doing something wrong. I'm messing up with the experiment. Looking at the southern plot, and there is this beautiful gene location signal, I also noticed that there is a very strong signal. There is a tiny bit like a dirt, another signal. And when I analyze the dirt, it is giving a signal from a human chromosome. How come? At that time, actually, the concept of pseudogenes was emerging new. I mean, the scientific community knew about the presence of pseudogenes. You know, there are processed pseudogenes and non-processed. All these details were very, 1,500 genes are known. So, I mean, pseudogene as a concept is there, but where is the pseudogene? Who is the pseudogene? I mean, maybe one or two examples. We made the conclusion that the real gene is like the dirt, and the strong signal is from the processed pseudogene. And since processed pseudogene integration into the human genome is random, it doesn't follow the groups of synteny rule. This was the insight, you know. This was the real critical point that we were at this very, very, very, very early stage made the connection with my mentor that there can be a pseudogene that tampers with the real gene, that hides the real gene. Today, 2024, I mean, we have about 25,000 genes, and how many pseudogenes we have?
Up to 50,000, depends.
50,000. Today is knowledge. But at that time, it was just a, hypothetical issue that people were discussing. So, I had no idea of going into pseudogenes. I had no idea of discovering Prader-Willi syndrome gene. I was investigating the comparative map of a juxtaposition of a physical map and a linkage map. And it is always in research, during the journey, when you encounter something unexpected, then you need to yorumlamak, neydi in English?
Interpretation.
Huh?
Interpretation.
You need to interpret it. And this is how it happens. The same thing happens another time. Let me share you some news with you. At the same time, while mapping these genes, I mapped the gene to human chromosome 17. And it was at the very point where this was a peripheral myelin protein gene. Peripheral myelin. Myelin is a sheet that insulates the axons. And the gene's it is the major protein of the myelin sheet. And linkage map already shows us that hereditary sensory neuropathy. This is the problem of the sensory nerves. We have nerve conduction velocity. A signal travels in the axons with a speed, I think 30 meters per millisecond. And the CMT patients, Charcot-Marie-Tooth patients, nerve conduction velocity is slow, below 20. And we wrote the paper. It was published in Nature in 1993, I think. And we mapped it to actually mouse chromosome. But looking at the synteny group, we speculated that it is on human chromosome 17 and is the cause of Charcot-Marie. Furthermore, signal intensity, while we were mapping, showed us that it may be 3 copies of the gene. Not 2 copies, but 3 copies. This was the first documentation of copy number variation in the human genome. Now, about 34 years later, recently, the American Society of Human Genetics, this is the largest organization in the world for genomics, did an analysis of the past 75 years in medicine and selected 6 discoveries. And one of them is discovery of CNV. Because now, today, we know that copy number variation is a major evolutionary mechanism. It is involved in many different diseases in humans and so on. So, you don't know what you're doing. Of course, you know what you're doing, but the implications of what you are doing, I had no idea that I would be defining a copy number variation in the human genome. There was no concept of copy number variation. We are diploid organisms. We have everything in 2 copies. Then here comes a concept of sometimes genes are in 3 copies, 4 copies. So,
I have to intervene. I'm sorry for intervening, but we may have some time restrictions.
Okay.
Maybe the security can come and kick us out, and maybe.
But I covered, I think, 90% of your questions.
Yes, you did a great job hocam. Teşekkürler. And maybe we can switch to Blitz Q&A session if it's okay for you. Okay, so
No,
maybe we can
no,
take
we
some.
can.
No, we promised to take questions from the audience. So to be able to do that, I think we shouldn't continue with our questions, but focus on the audience's questions, right?
Yeah. So we will proceed to the next Q&A session.
Remaining,
So anyone with any questions?
Okay.
So if anyone has questions, please raise your hand and ask, and I will start. I guess.
Yes, go ahead.
Hocam, at the beginning, you mentioned one of the problems was academic jealousy for
Yes.
you, and we are science in science fields. We sometimes also experience this kind of feelings, jealousy, especially compared to our peers who are more successful, maybe in some fields. How did you overcome it, or what's your advice to overcome it?
Your peers.
Yes,
Ignore.
Huh?
Ignore. Ignore. Issues. I use. Don't cry over them. And don't confront them either. Don't get into quarrel. But ignore.
What do you think about the future of our department? Like, do you have any plans to empower it?
My plan for the department. Let me first say that MBG is now last year also selected as the exemplary department of human genetics in the world, by America, not something else, but the Americans. So our whole purpose in the department is, of course, to conduct excellent research, because, again, I have to go into Turkish. Akli bilimler, nakli bilimler. Okay? İthal ikamet. I mean, you can get the information from outside and give the most recent information to the individual. Some people even think that this is the ideal model for education. At Bilkent, we happen to think a place where information is produced is the place to learn the best. Actually, from a philosophical point of view, Bilkent. Bil stands for something, Kent stands for another thing. Bil here stands for
Science.
science, bilim. Then some people came and said, no. Bil stands for bilgi, knowledge. Bilkent is not city of science, Bilkent is city of knowledge. What is the difference between knowledge and science? Yes.
One is storing something.
One is
storing information, another is producing.
I think you answered it perfectly well. One is static, the other one is dynamic. It is producing the information versus storing in an encyclopedia. So, for the future, I am hoping that the whole Faculty of Science will continue to fulfill this mission in our country, including the MBG. And this can be achieved only by producing people who are looking at the problems with critical thinking. and creativity, a creative mind. So my hope for the department, for the future, to be at the forefront of science by producing science from here that is exemplary to the world, leading the world, and also relaying this information to the young minds like yourself. This is what I hope for the future.
Questions?
As
Hocam, you asked us several times, but now I would like to ask you. Do you think this year's Nobel Prize was appropriate?
I think it is certainly appropriate. This year's Nobel Prizes certainly appropriate. Which brings the question, what happened 20 years ago? I think that's also very good research, excellent research at the same time. But the groundbreaking discovery was this year's Nobel Prize, for sure. So, my honest assessment of the situation is like it's been awarded, not exactly, of course, but repeatedly. You know, some people also say, look, cholesterol molecule has been decorated with, I think, 8 or 9 Nobel Prizes. So it is not unusual. MicroRNA is something so fundamental that it may get 2 Nobel Prizes. This is another argument. But when you look at 2006 Nobel Prize cites 2024 paper in the justification, and then 20 years later, 18 years later, that citation in the prize is the prize, then this is the critical. But on the other hand, it's also a very bold move, because everyone can make a mistake. It can be a grave mistake. It can be a and to face them is also another very mature, I think, thing to do. There are Nobel Prizes that are entirely, missed, lobotomy, removing the brain of people. I mean, it is terrible. It's a form of torture, but it was seen as a revolutionary treatment, and insanity is cured by, actually, insanity is cured by making it people vegetables. So, yes, you are curing insanity, yes.
Questions?
Yes.
Especially in today's age and today's generation in Turkey, I feel like everyone focuses on going abroad, staying abroad, living abroad, being as far away from home as possible. And you saying that coming to Turkey was one of the best things I've ever done, I'm wondering what drove you to this decision and why you choose to still live in Turkey when so many people today are against it.
I have my roots here. I love my country. I have my roots here. And so I'm not for young. I know all the difficulties. I know all the difficulties. I'm aware of them. But still, I'm also happy here. I mean, Mary Claire King, when she came to visit us, together with Diane Donnine, these are the, I think probably, Mary Claire King Have you seen any of her videos? She's the person who discovered BRCA1, breast cancer gene. A very actually active scientist, both politically and scientifically. She's a Lasker awardee. For the past 10 years, she's in the shortlist for Nobel Prize because of the discovery of BRCA1. Diane Dunlap is the best ever, I can say, I think, dysmorphologist that ever was trained and became a professor in Europe. They came here, and the final verdict they told me is, I mean, with Mary Claire's expressive personality, let me try to capture that. She said, Tayfun, you have everything here, and you have 10,000 years of history on top of that. Shut up. Bacaklarını kırarım. You spoiled boy. She's elderly. This is also the case. And nowadays, it is changing. I mean, with the availability of the internet, traveling all around the world, with all the connections that we can establish. Here also, the important thing is, I tell this to my colleagues, you know, in spring, I gave a lecture about this abroad and career advices. And I said, there are 5 diseases that you should avoid. One is I made these up. One is Gibraltar syndrome, Jebel Tarık syndrome, which is, you know the history of Spain when Arabs armies, Tarık, passed the Gibraltar Strait and ventured into the Iberian Peninsula. He burned all the ships so that his soldiers don't go back, escape. So, you guys should be like güdümlü füze, targeted missile. Once you lock with your target, don't look back. Ah, Avrupa'da olsaydım neler olur falan filan. Not good. Jodie Foster. You know Jodie Foster. In 2013, she got the Cecil DeMille Career Achievement Award. And this is a big award. In the award acceptance speech, word leaked to the press that she is going to announce she is gay. So she denied all her career. Then she is on stage. She gave her speech, and then and now I am. No one came. Everybody. I am single. And the camera Now, it took her a lifetime, a career achievement award, to admit the truth. Be true to yourself. In science, let's say you are in a field A. I've seen many people complaining, kör talihim, kara bahtım. I'm in neuroscience. However, I have the best mind to become an immunologist. And I am stuck in this field. Don't try to find excuses of this type. And whatever you are doing, do it. Yerinde, yeninde. Oynayın, gelin oynayın. Number 3. Ümmü Gülsüm syndrome. I think I told them. You know Ümmü Gülsüm, the legendary Arab singer. She is not anymore alive. Now, she has a phenomenal voice. You can listen to her on the YouTube. Imagine, she is performing in a concert and she has a rock band behind her. Dıms, dıms, dıms, dıms. O da ya Habibi söylüyor. I mean, it doesn't matter whether she has a beautiful voice or not. With that kind of an orchestra behind you, I doubt. I mean, still it would be the beautiful voice. Would you get the taste? On the opposite side, let's say Freddie Mercury is singing his and sazayeti. No. This means that whatever research project you're choosing, don't do it like a blind that sticks, to a stick. Kör değneğini bellemiş gibi. Always look at the situation from all the angles. Throughout my career, I changed gears. At Stanford, I was every night and day doing cell culture experiments. When I came back to Turkey, I said, no, this is a big mistake. If I start developing, it would be a strategic mistake. If I start developing, asking questions, projects that require cell culturing, then I'll be doomed. Because it's so difficult to handle these organisms. Is the electricity perfect? Is the water clean enough? There are all these factors. Imagine 30 years ago it was even worse. Now it's different here. So I said no. I'm now analyzing only the DNA molecule. The central point is DNA. It's a stable one. Plus, from a scientific point of view, we are, of course, a paradise for discovering the disease genes because of our family structure and consanguineous families. Then, in 2010, the game all started changing. Now we have big data and bioinformatics. Today, Barış Kerem knows very well. We are out of the lab. Not anymore. And we are asking questions that are relevant today with the latest tools. Still still the basic concepts don't change. But you have to take all of these into account. Next, Don Quixote syndrome. I mean, calculate, you have a mind, you have a brain, and don't run after the windmills. I mean, there are questions that can be asked, there are questions that cannot be asked. So, always take into account. And from a career perspective, always differentiate the difference between disruptive personality disorder and dystemic personality disorder. A disruptive person can be friendly, may seem, helping you and so on. But that disruptive personality may be really hard. On the other hand, a dystemic person is an unhappy person. And when you are next to him or her, it's a dark world and may criticize you all the time and make life, terrible. But that dystemic people may be telling some things to you that may help you. Don't judge him because he is dystemic and he is not pleasant. Okay? These are the things that I want to tell you.
Okay, hocam. I guess we can kind of cover up and shortly finish our meeting. Just few very quick questions.
Yes, no?
5 or 6. Yeah. What is your favorite gene?
I think I should say cryptochrome.
Cryptochrome.
Why?
Because the collaboration is no flight bullets.
Yes, and also because in the future, I hope it will lead to some more insights into the disease biology.
Okay. Which disease you hate the most?
I hate, of course, any type of cancer, and I hate psychiatric disorders because they are episodic, they are unpredictable, very difficult.
If you had a chance to be, you are already a very famous scientist, but if you had a chance to be like all known by the whole world, the most famous scientist in the world, with all papers in science journals and nature journals, for your work you are doing in the lab, but which will not be important after 10 years or 20 years or after you pass away, or if you work on the project which is unknown and no one recognizes it right now, but after you passed away, after 100 years, it will be a breakthrough. Which one would you choose?
I mean, of course, long-term thinking is the second option. I mean, nobody takes care of it, but takes it seriously, but 100 years later, after I pass away, it's a great discovery.
Okay. Teaching or research?
Benim yar, sadık yarım karam. Ne demek? Yeah, I think it is the lifeline is the research.
Your favorite song?
There are many. I'm listening
To do research at the same time.
Oh, for example, in the lab, I love to listen at that time, Madonna. At that time, yeah. I also, at that time, I liked to listen Sezen Aksu very much. I remember, I I was next. Yağdır Mevlam su. You know that song? A little bit arabesk. Sultan Süleyman sözünü mesela hatırlıyorum yavrularını. I remember. Last night we drank in San Pedro. Ya Isla Bonita. You know these songs? Yeah. So that's I always love classical music, but I mean it's not. I like it.
And lastly, your favorite Turkish food?
Turkish food. Oh, whatever my wife cooks. One thing. You ask this question, hayatta kocanı iyi seçeceksin. Karılı kocanı değil. And also, happy wife, happy life.
Okay, let's take a